Hello, this is Professor Young, and this is part 2 of 3 of our look at muscle physiology, where in the first session we spent some time kind of reviewing the 3 basic types of muscle, of course that being smooth, skeletal, and cardiac. We went over the basic anatomy of skeletal muscle specifically, and we spent a lot of time really talking about how the sarcoplasmic reticulum does what it does, which, as we have learned, is really central to the ability of skeletal muscle to do what it does, which is contract. And in this session, we're going to be spending a great deal of time going through and looking at step by step the process by which skeletal muscle is going to contract and the means by which it will also relax. And again, we're talking about deep down within the myofibers, down into the myofibrils, down into those myofilaments that we talked about last time. And remember, it is within those myofilaments where we see the physical act of contraction happening. And so it's probably a good place to start there, since on the screen right in front of us, there is that term staring us right in the face, that term being myofilament. For those of you who are lost, it's right there. And notice that there are really 2 types of myofilaments that we need to consider. We have thick myofilaments and we have thin myofilaments. Each of these have their own specific anatomy, and the anatomy is specific to the function that they perform during the act of contraction. Now, let me just kind of pause here for a second and make sure that we're all on the same page here. Notice that I said this is the process of contraction for skeletal muscle. Smooth muscle and cardiac muscle are going to deviate from the process that we're going to be learning about now. And so this here is specifically dealing with and looking at skeletal muscle. And so again, we have within skeletal muscle, we have these myofilaments and 2 types, both thick and thin myofilaments. What you are looking at currently is thick myofilaments. And in fact, if you pay attention here, and I'm talking about specifically this image right here, this is typically what a thick myofilament looks like. It's made up of the protein myosin, and myosin specifically here is located, all of these little punching bag looking things right here. These are all myosin, what we call myosin heads. So coming off of this protein, there are these extensions that are actually perfectly illustrated right here. And you'll notice that those little extensions that I referred to earlier as what appears to be punching bags have 2 components to them. They have a tail, and they have what we refer to as a sidearm or myosin head. And in fact, it's that myosin sidearm or that myosin head that goes through and will go ahead and cause the physical act of contraction within the interaction between the myosin sidearm and the myosin, within the thick myofilament and the thin myofilament. We're going to learn more about this interaction here in a couple of minutes. But for right now, know the structure. Right? So within thick myofilaments, we have the key protein here is myosin. It's got these extensions that are coming off of it, which is known as the myosin sidearm, or the myosin head, which is connected to the myosin tail. The other structure I will kind of bring you into the fold here with is right there, that little kink that kind of connects the sidearm to the tail, and that's a hinge. And that's what allows the myosin head to flex or move forward. So in the position that it's in right now, it's what we refer to as the initial starting position. And what will happen, it'll flex inward when it's appropriate. And we'll go through to learn what those circumstances are by which it's going to go through and do that. Thin myofilaments, which is what you are looking at right here. This is a little bit more detailed. Right? Notice there are more parts that make up the thin myofilaments as what we saw within the thick myofilaments. And so that's a big key distinction here that we need to not lose sight of. Right? The fact that the thin myofilament here is more complex overall than the thick myofilament. And so if we look here, we will see that the thin myofilaments is actually made up of 3 separate and different proteins. The main protein that we are concerned with is tropomyosin. And the reason why we say that's the main protein, the reason why we say that's the main protein here is because that's considered the backbone. When tropomyosin is in the inactive state, muscle contraction is not going to occur. Right? And so that's number one. When tropomyosin is in the resting state, action or muscle contraction is not going to occur. And so you can see right here in this picture, that rope-looking structure, that is the tropomyosin. Now, circling around tropomyosin is another protein. And that protein is called troponin. And again, troponin almost acts as a bracelet or an anklet surrounding tropomyosin. And you can see here in the yellow, that's a troponin. This is a troponin. There's a troponin. Right? And so those guys there, they are all troponins. Now, here's the job of troponin. Troponin accepts the calcium. Where's the calcium coming from? It's the calcium that was released from the terminal cisterns associated with the sarcoplasmic reticulum. Right? There's the connection for what we learned last lecture. All right. When we look at muscle contraction, we know that the calcium is released from the terminal cisterns, and the trigger for that release comes from the action potential that has moved down the sarcolemma down into the T-tubule. The trigger for that action potential, remember, was the release of acetylcholine down into the sarcolemma. Acetylcholine was released from the neuron into the neuromuscular junction. The acetylcholine goes down, it binds and allows for the sodium gates to open up. Sodium is released, which causes the action potential. The action potential travels down to the next sodium gate, which opens that up. Travels down to the next sodium gate, next to the sodium gate, next to the next sodium gate, until that action potential reaches reaches the voltage-gated calcium channels associated with the terminal cisterns, at which point in time those channels open up, it releases the calcium goes into the sarcoplasm, and it is here where the calcium finds its task. It binds to that troponin, and when calcium is bound to troponin, troponin forces tropomyosin to spin. And when it spins, it uncovers the third protein associated with thin myofilaments, and that is actin. Now, once actin is uncovered, it can then go through and bind to myosin. Remember, myosin is the protein that is associated with a thick myofilament. All right, so that's how this is all playing together, and it's all playing very nicely together as we look at things. So, associated with the thin myofilaments are three proteins: tropomyosin, which makes up the backbone. When it's in the inactive state, muscle contraction does not happen. When it's active, muscle contraction is allowed to happen. Why is it allowed to happen? Because troponin, that being the second protein, binds to the calcium, which forces the tropomyosin to spin. And when tropomyosin spins, it uncovers the actin. And when actin is uncovered, it can bind to the myosin. Makes sense? Hopefully. Good. Let's move on. Let's stop here real quick and just think about the overall structure that we are really referring to when we talk about thick and thin myofilaments. All right, I'm going to show you an image here in a couple minutes that is going to show you the orientation of thick and thin myofilaments. Within that orientation, we have places where there is only thin myofilaments, we have locations where there's only thick myofilaments, and we have locations where there is an overlapping of thick and thin myofilaments. This slide here is defining those regions for us. All right? And so when we have an area that is both thick and thin myofilaments present, we call that the A band. Thick and thin myofilaments are overlapping one another. They are both present. We refer to that as A bands. Now, when there is only thin myofilaments, then we refer to that as, sorry about that, we refer to that as I bands. Thin myofilaments only, and areas of thick myofilaments only are referred to as H bands. A bands are both thick and thin. I bands are only thin. H bands or H zones are only thick myofilaments. When these thick and the thin myofilaments overlap one another, that is when we see striations within the muscle. So go back to the histology and think about those striations. What you're really seeing coming through the myofiber is two layers down. You're not seeing the myofibril, you're seeing the myofilaments shining through those various layers as the thick and the thin myofilaments are overlapping one another. One complete set of thick and thin myofilaments, including the A bands, the I bands, the H zones, the Z lines, the M lines, and everything else. One complete set of thick and thin myofilament is what we refer to as a sarcomere. One complete set of both the thick and the thin myofilaments is what we refer to as a sarcomere. And so we can see that here on this slide. That's the slide that I was just talking about. All right. So again, one complete set. This here is one complete set. So that would be one sarcomere. This here would be a second sarcomere right there. And this here would be yet a third sarcomere right there. And so here you're looking at three distinct sarcomeres, or three complete sets of thin myofilaments and thick myofilaments. Again, you can see the A band, which is both the thick and the thin myofilaments. That I band there is only thin myofilaments, and the H zone there is the thick myofilaments only. All right, so be familiar with this overall setup. All right, be familiar with that overall setup. All right, what is about to happen next is what we refer to as the sliding filament theory. I have given you the key steps involved in muscle contraction. Key steps without some minor details added in. And what we're going to go through now and do is fill in those minor details. So far, you should be able to explain the fact that a neuron is going to innervate a sarcoplasma, or a nerve is going to go ahead and make contact with a muscle fiber, and that muscle fiber, I should say, that interaction is going to occur what we refer to as a neuromuscular junction, where acetylcholine is going to be released into the synaptic end of the neuron. It's going to diffuse across a space down into the muscle sarcolemma, into the muscle membrane. That acetylcholine is going to come and drift down into the tissue. It's going to go ahead and spark the opening of a sodium channel. That sodium is going to be released into the sarcoplasmic reticulum. All right, and so that sodium goes ahead. Once it's opened and released, it causes an action potential. That action potential travels to the next sodium channel, which is going to cause sodium to rush out, which is going to send that action potential down to the next sodium channel. It's going to open, sodium is going to rush out. It's going to send that action potential down to the next one. That's going to continue until that action potential reaches the terminal cistern. It's associated with the sarcoplasmic reticulum down into the T-tubule, at which point in time that action potential is going to go ahead and force the opening of the voltage-gated calcium channels, releasing calcium into the sarcoplasm, at which point in time that calcium binds with troponin, which is going to spin tropomyosin, uncovering the actin, allowing actin and myosin to bind, and forcing a contraction. Right? So far, that's where your understanding should be. That's what you need to be able to understand. By the time we get done these 5 nice little neat steps that are going to outline for you action potential, you're going to be able to plug in the rest of the detail. And remember, it's always about the detail here. So why don't we go ahead and get ourselves started with looking at what here is left to fill in for muscle contraction. And again, this is going to take place over 5 steps. But what you also need to keep in mind here is that before we go to step 1, we need to look at step 0.5. And actually, this is going to turn out to be step 5.5 before the cycle starts over again. And I'll point that out to you at the end of this. But what ends up happening before anything else occurs, A, there is no calcium present within the sarcoplasm. All right. There's no calcium present within the sarcoplasm. We know that this here is important because whenever calcium is present within the sarcoplasm, we have contraction. This is before contraction begins. And so what that implies is there is no contraction happening. The only way that happens is if calcium is completely removed from the sarcoplasm. So that's number one, right? There's no calcium present within the sarcoplasm. Number two, the other thing that now also has to be true is ATP would have bound to the myosin, and it would have created ADP plus a phosphate. So we have ATP that's bound to the myosin. It's released energy in the form of ADP and that third phosphate. There's no calcium present within the sarcoplasm. We are now ready to officially begin step one. All right, now, a lot of this is going to sound very familiar to you because remember, we're just going back in here and filling in the details. All right, so here are the details. Number one, for step one, we have neurotransmitters that are released. You already know that the transmitter that we are referring to is acetylcholine. That's the transmitter of muscular contraction. So acetylcholine is released into the muscle cell. That acetylcholine, that neurotransmitter, allows the sodium gates to open, which begins an action potential. That action potential is going to continue to travel around the sarcoplasmic reticulum until it reaches the voltage-gated calcium channels associated with the terminal cisterns, at which point in time calcium is released into the sarcoplasm and it binds to troponin. There is nothing new there. This is something that we have gone through. I have drilled this into you. I have stressed upon you the fact that you need to be able to explain that. You have step one down, no problem. In fact, you even have step two down with no problem. Step two is the calcium binds to the troponin. Once calcium is bound to troponin, it forces the spinning of tropomyosin, and the actin is uncovered. And we know that once actin is uncovered, actin can bind to the myosin. Now, here is something that we did not go over yet. Actin and myosin don't just magically bind to one another. That doesn't happen. In fact, how actin and myosin bind to one another is all dependent upon ATP. ATP must first bind to myosin, and then the actin that is uncovered can bind to the myosin. We got that? We good. Because if we ain't good, we can't move on. Acetylcholine is released into the neuromuscular junction from the neuron down into the sarcolemma. From there, the acetylcholine goes down. It comes down until it activates and opens up the sodium channels around the sarcoplasmic reticulum, which allows the action potential to begin to travel around that sarcoplasmic reticulum, coming to sodium channel after sodium channel after sodium channel. That action potential continues down and down into the T tubules, where it will go ahead and activate the voltage-gated calcium channels, releasing the calcium into the sarcoplasm. binding to the troponin, spinning the tropomyosin, uncovering the actin, and in step 2, ATP binds to the myosin, which will allow the actin and the myosin to bond. If you don't have that down just like I did it, then you need to stop this presentation and rewind and make sure you can say it with me until you can say it by yourself before you move on. Don't try to learn this whole complicated minutia in one full swoop. Break it down. Chunk it. When you chunk it, you're going to learn it in bits and pieces, and you'll be able to connect it much easier. Step 3. This is what we refer to as the power stroke. Why? Because this is where the myosin head attached to that hinge springs forward. And when it springs forward, it pulls the thin myofilaments in towards that Z line, that middle pole, the anchoring pole that anchors the thick myofilaments down. Those thin myofilaments come in. And so the myosin sidearms turn so that the thin and the thick myofilaments slide past each other. The sarcomere shortens, and when the sarcomere shortens, the myofibrils shorten, and when the myofibrils shorten, the myofibers shorten, and when the myofibers shorten, you've got yourself muscle contraction. You have yourself muscle contraction. That takes energy. Notice, in step 2, I said nothing about ATP releasing energy. The only thing I said was that ATP must bind to myosin before actin will bind to myosin. It is in step 3 where that ATP that was bound in step 2 releases, cleaves. That ATP releasing the phosphate group and leaving behind ADP. That's the energy. The releasing of that phosphate group is the release of energy that allows myosin to spring forward, forcing the actin and the thin myofilaments in towards the center, causing contraction. We're done. It's over. Wipe your hands clean. Contraction has happened. But we're not done yet. You can't quite just go ahead and forget about the rest of the process. Because at some point in time, we need to reset this whole thing. And this is what happens in steps 4 and 5. It's resetting the process. A new ATP comes in and binds to the myosin. When that ATP binds to the myosin, it lets go of the actin. It lets go of the actin. And then it is in step 5 where the myosin will go ahead and release a phosphate group from the ATP, creating ADP, and the myosin head resets itself. It goes back into the starting position, waiting for actin. When would actin bind to myosin again? If calcium is still present in the sarcoplasm. Why would calcium still be present within the sarcoplasm? Because an action potential has moved along the sarcoplasmic reticulum, triggering the voltage-gated calcium channels of the terminal cistern to open back up, releasing more calcium. As long as the action potential is coming down, traveling along that sarcoplasmic reticulum down into the T-tubule, calcium is going to be released. And as long as that calcium is released, it can combine to the troponin, which is going to spin the tropomyosin, which is going to leave actin uncovered. And as long as actin is uncovered and there's ATP available, myosin and actin will go ahead and bond. And as long as actin and myosin are bound in the presence of ATP, contraction will continue. As soon as the action potential stops, calcium will be returned to the terminal cisterns through the use of the calcium pump, which we know requires ATP. The calcium pump is pumping excess calcium from the sarcoplasm back into the terminal cistern. And as soon as you remove that calcium from the sarcoplasm, there is no longer calcium that is present to bind to the troponin, which means tropomyosin is going to return to the blocking stage, if you would. And as soon as it returns to the blocking stage, actin is covered. It's not allowed to bind to the myosin. The contraction is done. What determines whether or not an action potential continues? I'm sorry, I lied. What continues? Well, what determines whether or not a muscle contraction continues is the presence or the lack of the action potential. The action potential is what controls everything here. Be very clear about that. Be very clear about that. Now, you may be feeling just like this right now. And if you are, that is okay. I give you permission to feel this way. But not for long. Because the bottom line is you need to be able to explain clearly how we run an action potential. When the ATP binds, when phosphate groups are released, when calcium is present, when calcium is not present, who's binding to who, what proteins are associated with which myofilaments. You need to know this like you know the back of your hand. In case you have forgotten, and I'm going to leave you on this note, but in case you have forgotten, this is the complete story, starting in the middle and working to the end. Acetylcholine is released within the neural junction, that neural space. It diffuses across. That acetylcholine is going to diffuse across down into the sarcolemma, where it's going to penetrate into the sarcolemma. It's going to force the opening of sodium channels down within the myofibrils. The release of that sodium is going to spark an action potential, which is going to travel along the sarcoplasmic reticulum. It's going to go, that action potential is going to travel to another sodium gate, which is going to open up the gate, allowing sodium to rush in. Boom! Propelling that action potential to the next sodium gate, where it's going to open up that sodium gate. Sodium's going to come in. Boom! It's going to propel that action potential down to the next sodium gate, 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 until that action potential reaches the terminal cistern associated with the sarcoplasmic reticulum, down deep within the T tubule, at which point in time the voltage-gated calcium channels are going to open up, releasing the calcium down into the sarcoplasm. At that point in time, the calcium that is now present within the sarcoplasm can bind to the troponin to spin the tropomyosin to uncover the actin, but before the actin can bind to the myosin, ATP binds to the myosin, allowing the myosin and the actin to go ahead and join into one. At that point in time, the myosin is going to go ahead. It's going to have the ATP release energy, freeing, cleaving, breaking apart that phosphate group, which is going to go ahead and force the myosin to spring itself forward, which is going to pull on the actin attached to the thin myofilaments, causing a shortening of the sarcomere. Shortening of the sarcomere means a shortening of the myofibrils. A shortening of the myofibrils means a shortening of the myofiber. You've now witnessed a contraction at step 3. Step 4 and step 5 of the sliding filament theory is now going to be focused on resetting the myosin head for the next actin to come in and bind to it. And so a new ATP molecule binds to the myosin, releasing the actin. And the myosin is going to cleave that ATP, releasing the phosphate group, allowing the myosin head to reset back into its initial position, and assuming that another action potential has traveled all the way down into the T tubule, releasing more calcium from the voltage-gated calcium channels within the terminal cisterns. If that has happened, then there's more calcium to bind to the troponin to keep the tropomyosin spun, uncovering the actin. Another ATP, of course, would then come in and bind itself to the myosin, allowing the actin and the myosin to go ahead and stay bound together, which is going to perpetuate this contractional force that is happening. But, wait for it, if the action potential has ceased to travel down the sarcoplasmic reticulum, down into the T tubule, therefore preventing the release of calcium, and instead of releasing calcium, now the terminal cisterns is activating the calcium pumps again, using ATP, but it's going to remove the calcium from within the sarcoplasm, which means no calcium to bind to the troponin, which means tropomyosin spins back on itself again, which means actin is covered, which means it is blocked from binding to myosin, thus ending the muscle contraction. And that is what you need to be able to explain. I will leave you now to go ahead and continue reviewing this process.